We have Kim Taylor to present on surveying for Texas Screwstem or Bartonia paniculata subspecies texana in East Texas, or on the trail for Texas Screwstem, elusive, tiny, screwy. Kim Taylor is the manager of the Conservation Seed Bank at the Botanical Research Institute of Texas and Fort Worth Botanic Garden. Kim joined Britt in 2010 following completion of her master's degree in botany from Austin Peay State University. Kim took over management of the newly opened Conservation Seed Bank in 2020. Her main focus is on research and conservation of rare Texas plants and ecosystems. As seed bank manager, she conducts rare plant surveys, tracks and monitors populations of rare plants in North Texas, collects seed of rare species to preserve genetic diversity, and works with partner institutions to fight plant extinction in Texas. Thanks, Kim, and it's all you.
Thank you, Anna. Hi, everybody. Thanks again for joining us on this second attempt to do this webinar. Our first attempt, of course, was during our winter weather last month. So I'm glad that you all are here today. Before we get started here, I just want to share a little bit about kind of who I am and what I do, in addition to what Anna has just shared. So I am the seed bank manager at for the conservation program at the Fort Worth Botanic Garden and Botanical Research Institute of Texas in Fort Worth. And the goal of our conservation program is really to prevent plant extinction in Texas. And we do that in a number of ways by surveying for rare plant populations, studying rare plant biology, tracking populations, in particular of rare plants in the Dallas-Fort Worth Metroplex area, so monitoring those populations on a regular basis. And then collecting seeds for these rare species to store those as a genetic backup in case the worst comes to worst and we do lose some of these populations. So when Anna approached me about this project to look at Bartonia, I was really excited because it fit in really well with our goals of looking at these rare species and trying to understand really where they grow and what their future might look like. So what is Texas Screwstem? Texas Screwstem, or the scientific name is Bartonia paniculata, subspecies texana. It's a small member of the Gentian family. It is an annual species. It is a very small plant, and you can see it's mostly green here. It is only about maybe 6 or 7 inches, would be about the typical height of a plant. It's a single stem, basically almost no leaves are visible on the plant. So it's a very difficult plant to see in the field. It was first collected in 1965 by Donovan Carell. And in 1966, he described this specimen that you see here as a new species, as Bartonia texana. He thought that it looked different from a very similar looking and closely related species, Bartonia paniculata. But he noted some differences and described it as a new species. So he described it in 1966, and if we kind of look at a just overall timeline of what's happened with this species, each of the little leaf pieces on this timeline represent a year that the plant was seen in the wild, either at one population or at multiple populations. So you can see that it hasn't been observed all that frequently throughout its history. First observed in 1965 and described in 1966. A few populations found after that, and then in 1980, Nixon and Ward did an in-depth survey for this species, and they were able to find a number of new populations, bringing the total number of populations up to 11 populations. 1988, Orzel and Bridges did some more surveys. We're now up to 14 known populations. And that was really, in 1988, the last major attempt to look at the distribution-wide range of the species, to try to look for a number of new populations and try to update the status of the species. So that brings us all the way up to 2018, which is when this study that I'm talking about today started. And at this point, we're up to about 22 populations in the state of Texas. Now, I do want to note that in 2009, something rather important for this plant happened. There was a paper published by Matthews et al., which took this species that was at the time called Bartonia texana and reduced it to a subspecies. And what that means is that Matthews et al. Decided that the that Bartonia texana was probably more closely genetically related to this Bartonia paniculata species and some other subspecies within that instead of being distinct. So we reduced it to a subspecies, and there's still some taxonomic uncertainty on this species. Is this a good species? And so that is a legitimate question, and a question that anybody dealing with this species has to grapple with, and that's understanding if we really are dealing with a legitimate species here. And you might wonder why that's important. It's important because if we are talking about doing conservation work for a species that is not actually a good species, it's not genetically distinct, then we're wasting our resources. So it's really important to answer this question. And I think that as of right now, I work under the assumption that this is a good species, or at least a good subspecies. It is a unique entity, but I think there still needs to be some work done on this. So for the sake of this survey, we're assuming that we are dealing with a unique entity, but that question still needs to be addressed. Okay, so let's look at the distribution for this species. Bartonia occurs in eastern Texas in about 10 counties and 3 counties in Louisiana. This is the worldwide distribution for the species. It only occurs in this very limited geographic area, which is one of the reasons why the NatureServe conservation status rank for the species is a G2, or globally imperiled. And the NatureServe conservation status ranking system basically ranks species from a scale of 1 to 5, where 1 would be critically imperiled. These are the species that are at a very high risk of extinction. A G5, or secure, would be your common species, your widespread species, species that are at very low risk of extinction. So at a G2, or imperiled, Bartonia is at a high risk of extinction or collapse due to restricted range, which we see with this species, a few populations or occurrences, which we see with this species, and then potentially due to steep decline, severe threats, or other factors. So we're a G2 in the state of Texas. Looking at just the Texas populations, we also have a ranking of 2, or S2. And in the state of Louisiana, it has a critically imperiled ranking, since there's only, I believe, 3 populations in the state of Louisiana. So that's an S1 ranking. So for this presentation, I'm going to be focusing on the Texas populations. Unfortunately, we won't be talking much about these Louisiana populations. So NatureServe is a really great method to track our rare species and understand their vulnerabilities. But there's really no regulatory mechanisms that come from the NatureServe ranking. So we can give something a NatureServe rank and help us understand how common it is, or how at risk it is, but it doesn't have any regulation behind it. There's no way for us to actually protect those species at the federal level. To do that, we have to turn to the Endangered Species Act. Now, Bartonia has been present with the Endangered Species Act essentially since the Endangered Species Act was signed into law. In 1980, Bartonia paniculata subspecies texana was designated as a Category 1 candidate species, meaning that it was seen to be warranted listing. At that time, there was only known from a couple of populations. But after those surveys by Nixon and Ward, where they found a number of new populations, that designation was changed in 1983, and it was redesignated as a 3C candidate species. And that basically means that because all that new information was found for the species, it was decided that it wasn't a priority to list that species. So it was removed from the list. In 2010, it was petitioned once again. And in 2011, the U.S. Fish and Wildlife Service published the 90-day finding that said that, yes, this species may be warranted due to all of the reasons that they look at. It seemed like that this species is at risk enough to be potentially listed as a threatened or endangered species. So where we are right now is we're in this point of a status review and peer review in preparation for the 12-month finding. This 12-month finding would be where the Fish and Wildlife Service would propose whether or not the species would be petitioned or would be listed as threatened or endangered. So we're at this point where we're gathering information and prepping for all of this. So 2023 is the target year for this 12-month finding. So we'll know fairly soon whether or not this species will be listed. And this project really comes out of this need to understand the distribution for the species. So our project goals were really to consolidate the location information for all of the known populations of this plant in Texas. The Texas Conservation Action Plan put out as one of its priorities to consolidate information for rare species. In particular for plants, that's really important because there are almost 450 species of greatest conservation need, which are species that are at risk within the state. And we only have a handful of botanists working on these species. So there's a lot of information out there, but there hasn't been a lot of or enough effort to bring all that information together. Information is coming out quicker than we can consolidate it and compile it and analyze it. So our first goal was really to do that and get all the information about the species into one place so that we could start to look at its status and what it's actually looking like in the field. Our second goal was to then head out into the field and try to relocate and revisit the known sites and try to update that status. We have a number of populations for this species that had only been seen once or hadn't been seen in 50 years. So we really wanted to go out and try to go back to all of these locations where this plant was seen and see what the status was. Okay, so let's talk about where Bartonia occurs. I was really excited when we started this project because I had been in these habitats called Bagels, but I didn't really know much about them and I hadn't gotten to do a lot of field work in them. And it's just a really cool environment to be in. So Bartonia occurs in this habitat called Bagels or Seepage Slopes. And these communities are very densely wooded, as you can see in this photograph, and they will typically have some sort of impenetrable layer in the subsurface, like a clay pan layer, for instance, something that prevents water from percolating down through the soil. So we'll have water sitting in these areas either permanently or semi-permanently. It's often very slow moving or stagnant water that's accumulating in these areas. The soils in these areas are very acidic. We did some soil measurements and I believe it was a pH of about 4. So it's a very acidic pH for these species to occur in. There's low available nutrients, high organic matter, and a really interesting feature that you can see here. These are moss-covered hummocks. And what these are is basically soil accumulation around the root balls or bases of trees that is kind of covered with moss. And so we have this kind of microtopography within these Bagels where we have this really saturated standing water and then these little islands that are popping up out of those. And within the Bagels, it's really these little islands, these little moss-covered hummocks where we find Bartonia. So if we look at where Bagels are placed on the overall landscape, you can see here down at the bottom of the slope, we have a channel. This is some sort of waterway, a stream or a creek. And you'll find Bagels in association with these kind of surrounding these waterways. And then if we move up to the higher elevations, we have our more upland vegetation like our upland pine savannas. But the area in between, if we go from our high elevation to our low elevation, we have these seepage slopes. And what's happening is we have water that is hitting these higher elevations. It's hitting some sort of impenetrable layer and moving laterally across the landscape. So the water emerges out on these slopes. So we end up with these really moist slopes, and this water will eventually feed its way down into the Bagel. So you can see here in this center photograph, this green vegetation on the slope. This happens to be where we have moisture coming out, and you'll see an abundance of ferns and sedges and vegetation that typically likes moisture environments. So Bartonia within this habitat is going to occur in the seepage slopes here. So you can see that most of these photographs are of these slopes growing up out of the leaf litter. We have heavy leaf litter on the ground, a lot of magnolia leaves, for instance, growing on the ground. And then you can see some moss-covered hummocks. That's where Bartonia would occur as well. Associated species, the species that you see are really the key to knowing that you're in the right habitat. So we're looking for species like sweet bay magnolia as one of the dominant tree species, Bagel bush or Virginia sweetspire in the understory. So these are shrubby species that we see almost at every single site where we find Bartonia. And then down on the ground, the easiest thing to see, to point out that you're in the right place is just an abundance of ferns. You don't see these ferns in the drier environments. You see them where we have moisture. So they're a really good indicator of ferns. So netted chain fern is one example. And then of course we have our sphagnum moss. This is the moss that we find on most of those hummocks. And then there's two additional species that I want to point out. They're not dominant species, but they're really good indicator species. And that's northern blue thread or Bermannia biflora and nodding nixie, Apteria aphylla, the scientific name. And these are two little, very small plants, a couple of inches tall, but they grow in almost the exact same microhabitat as Bartonia. And they're a lot easier to see because they have some color to them, whereas Bartonia is just a very little green stem. So we found that these two species, one of them was present at pretty much every site where we found Bartonia. And anywhere where we found a really good high quality seepage slope or bagel, we found one or both of these species. So they are a really good indicator. And these two species actually serve to be a good way for us to ask people. We had a gentleman that was showing us another property, and he was particularly fond of this nodding nixie, and he told us about a place where he saw this at a property that we were going to, and told us where he saw this plant, and we were able to go there, and it led us directly to a Bartonia population. So these are really good indicator species. So the seepage swamp and bagels are a very narrowly distributed habitat. In the state of Texas, you can kind of see the distribution here in green of where we see that habitat occurring. And if we superimpose that with the distribution of Bartonia at the county level, you can see that Bartonia is basically found where we have this habitat occurring. So if we move on to kind of what we worked on for the project, our first step was really to get in there and try to identify the locations where plants were found. So we went to a number of sources to compile the location information for these plants. We turned to the Texas Natural Diversity Database, which is the keeper of the rare plant and animal location information for the state. And we got a really good start just by starting with what they had present. We also scoured the scientific literature looking for any new references to populations or to anything about the plant that someone has published somewhere that we hadn't seen yet. We also turned to the many herbaria in the state that have specimens collected by plant collectors, and we're really fortunate that this project coincided with a really big digitization effort to digitize these herbarium specimens. So we were able to get information about specimens that we wouldn't have been able to travel and visit those specimens. We were able to get the images, which was a really big help. And these herbarium specimens really are the primary source for location information for this species. It's not a big flashy species, so you don't see a lot of people making notes of it anywhere. You typically will just see specimens of it. And then finally, another really good resource is iNaturalist. So we turn to iNaturalist to find more updated information about populations. So if you're not familiar with iNaturalist, it is a community scientist platform where individuals can make observations of plants or animals or insects in the wild and upload those photographs and the location where they found them to this community platform. And as a conservationist, I can come in here and I can search for these species that I'm interested in and see all of the places where it was seen. Some of them I can see in real, you can see this in real time. So I can see things that were seen yesterday, and I can go out there and do work on that population. We had an instance a couple of years ago during the City Nature Challenge for iNaturalist where somebody found a new population of a rare plant in Dallas that we thought was completely removed from that county. We thought there were no more populations of that plant in that county. And somebody during that City Nature Challenge found a new population. So that was really exciting. So iNaturalist is a really great tool to help us find new populations. It wasn't as great of a tool for Bartonia because it's such an obscure little plant and hard to see. but there certainly were some observations by other botanists that gave us some good information. So after we pulled all of this information, our next step was to take all of these locations and put them onto a map. So if you look at an herbarium specimen and we pull up the label, you can see along with the name of the specimen, we're given a location description. And for many of these plants, these collections were made before GPS units became widely used. So this one was made in 1965. And we're given a description, along Clear Creek, 7.5 miles southeast of Colmsneil on Route 256. So this is the only location information that we're given. So we have to try to take this information and translate that into a location on a map so that we can try to revisit that spot. So we go to our trusty aerial imagery, often Google Earth. You try to figure out where on the map that might be. And then you look and say, okay, well, I'm not really super certain that this was exactly the location. So we add some sort of buffer around it. If we're given like an exact intersection, our buffer might be relatively small. But if we're given just a county, for instance, you know you have a really big area that plant could have been found in. But ultimately, that translates into a set of GPS coordinates and some sort of error range, so we know generally about how large of an area we need to go and search. So we did that for all of these, for all of the records that we found. We found a total of 50 unique observation records. So that could be a mentioning of the plant in a report. It could be an herbarium specimen or an iNaturalist observation. But those 50 unique records corresponded with 22 mapped populations. So a number of those records were of the same population, either at a different time, or it could have been a population where someone made a collection and then made a note of that in a report and then posted that on iNaturalist. You could have had several observations of the same point in time. Of those 22 mapped populations, 9 of those populations were not at that time present in the Texas Natural Diversity Database. Many of those populations that were not present were newer populations where the data just hadn't quite made it into the database, or they were things that we found in obscure reports or in small herbaria that had not been visited. So this really highlights that need, even for species that have had a lot of work done recently, to constantly be keeping up with keeping this information consolidated. 14 of those 22 populations had not been seen in over 30 years. So these are populations where nobody had seen plants at that location in over 30 years. And 3 of those, no one had seen plants in over 40 years. So some of these populations, we really have absolutely no idea what is going on. Are there still plants there? Or has the property been turned into a warehouse? We just don't know what's going on at that location. 17 of the populations had only been observed one time. So 17 of them, that's the majority of the populations. Somebody had found plants, maybe made a collection, and then no one had ever seen plants there again. So there's a lot of just unknowns about what's going on with this plant. 14 of the populations did not have GPS coordinates associated with them, so we had to try to pinpoint where the plants were. And 7 of those had such vague descriptions that we really have no idea where the plants were. It could have been a county or that was listed, or it could have been just a single unit within the Big Thicket National Preserve. So areas that are so large that we have no way of knowing for sure where they actually observed that plant. So this really highlights for those of you who collect data in the field to please make good observations of where you were. Okay, so we've consolidated all of our information. We know where these plants are. The next step is to head out into the field. So we did field work to revisit as many of these populations as we possibly could. In the fall of 2018, we were able to visit 14 of those previously known populations. Some of the populations were really recent, and we didn't want to waste the time on a population that had been seen a year before when we had older populations to go look at. And then some of the populations we were unable to get access to the properties. But 14 we were able to visit. Of those 14 populations, only 5 had plants present at the time of us visiting. In addition to those 5, we found 1 new population that had never been seen before. And across those 6 total populations, we only found a total of 64 plants. So that ranged from a population that had only 4 plants as the smallest population to a population that was as large as 22 plants. Okay, so really small populations. 4 of those populations were on public land, and 2 of those 6 were on private land. And if you look over here at the map, the populations where we found them were pretty much all in kind of the southern portion of the distribution of the species. So I want to just talk a little bit about some of the populations where we did find plants, because there's some interesting stories here. So the first place we found plants was the San Houston National Forest. This is in San Jacinto County, first observed by Eric Keith in 2006. And this was the largest population that we found. 22 plants we found at this population. And, the San Houston National Forest is an extensive area, and we only searched a very small portion. And, there's so much just really good, high quality habitat in this forest that I would not be surprised if somebody were to go out and find even more plants in this population. The second population, the Nature's Bottom and Jack Gore Bagel unit. This is in the Big Thicket National Preserve in Hardin County. It was first found during those 1980 surveys by Nixon and Ward. 11 plants were found in this population. Now this one is interesting. One, we found these in December. It was our very last day of field work. We didn't think we were going to find anything on that trip so late in the year, and we happened to find this population. So it was a really good end to the field work. But this population, the only reference we had to it was from a single herbarium specimen. And I put the label here for you so you can see what it said. It said northeast of Silsbee, Texas, within the Nature's Bottom and Jack Gore Bagel units of the Big Thicket National Preserve. It gave us no, location within that unit, really very little information. And if you're familiar with the Big Thicket at all, or with this particular unit, it's a rather extensive area. It is very difficult to access. There's very few roads. A lot of them are washed out, and very difficult terrain to get through, very few hiking trails. And all total, it's almost 14,000 acres. So we were really a little daunted when we decided to start looking for plants in this area. But we thought, the name is the Jack Gore Bagel, so surely there's a lot of good bagel habitat here. So we decided to search for this. And as we're hiking through some of the trails, we came across these signs. I just kind of posted along one of the trails. And it was for the Old Wagon Road Trail. And this was a trail that basically followed the route of this old wagon road that settlers used to use to traverse through the area. And apparently there were these springs along this trail that were typically used for people to stop over and get water. So we're walking, we see this, we're like, oh, okay, well, a spring, that might be a good starting point. So we went in search of these springs. And we ultimately found the springs. We didn't find any Bartonia at the springs, but we followed the water from there and were able to find just a beautiful seepage slope that you can see in this image here in a rather extensive, just really high quality habitat. And so that's how we were able to find this population. Now, I suspect that there are probably a number more plants in this unit. Who knows if we were in the same spot that Nixon and Ward found a population or not. But we're certainly really happy to have found that population. The next population that we found was at the Turkey Creek unit, also in the Big Thicket in Tyler County. Now this population was first seen in 1967, so it hadn't been seen in 51 years. And this is another population where the only reference we had to it was this rather vague description in a report from Geraldine Watson on the vegetation of the Big Thicket Preserve. So she wrote, one plant found in sphagnum at edge of savanna in Turkey Creek unit. So again, we're faced with this task of looking at this rather extensive, 8,000 acre unit and trying to pinpoint where we might find plants. So we were able to find plants in this unit, and again, a lot of really high quality habitat. And I suspect that while we may not have been at the original location for this plant, that there probably are more plants present in this unit. And then the final population I want to just pinpoint share a little information on is another big thicket population. This is the Big Sandy Creek population. This is the population that we found new. So this was a new discovery for us. So we were really excited on this one. And this one, there was in that same 1982 report by Geraldine Watson, she mentioned that Bartonia might be found in this unit, that it was possible that it could be in this unit. And presumably she said that because there's just so much really good high quality bagel habitat in the Big Sandy Creek unit. So this population, we followed a lead from someone who told us about that nodding nixie plant, and this is where how we were able to find this plant. We found 5 plants here, and again, I suspect there's probably a number of additional plants present. So there's the Big Sandy unit. And again, this is another rather extensive unit, about 14,000 acres. So there were 2 additional populations that we found plants at, both of those on private property. So I'm not going to go into too much detail on those. I'll just point out we had one population with 15 plants that hadn't been seen since 1986. And then another population where we only found 4 plants in Newton County that was first found in 2004. So it was really good to be able to confirm that plants were still present at this population that had been seen in 2004, and to be able to be there to help rediscover this population that hadn't been seen in 1986. So what about the 9 populations that we didn't find plants? So we visited 14, we found plants at a total of 6 populations, but there were 9 that we visited where we were not able to find any plants. So let's talk a little bit about what might be going on with those populations. The first thing I want to point out is that this plant is incredibly hard to find. It is a tiny little thing. It's a single green stalk, very few leaves, and you really have to be on top of it to be able to see it. So this image over here is a hummock that has 4 plants on it, and you really can't see the plant until you are right on top of it. Even when you're right on top of it, sometimes it's difficult to see. So if you can see the plant is right here in the middle. So I certainly do not feel confident in saying that these populations that we didn't find plants don't actually have any plants. It's very, very possible that we missed a lot of plants, that we did not happen to look at the hummock that had all of the plants on them, because most of these populations, we only found plants on a single hummock or on maybe a handful of hummocks in close proximity to each other. So if you just happen to not look closely at the one hummock that had these plants on it, you can miss an entire population. So I would never say that the plants are not present at any of these sites because it's just so difficult to see. Another thing that could have been going on is that for some of these populations, we had really poor location information, right? We didn't have GPS coordinates. We had a somewhat vague description of the location. So we did our best to try to locate where we thought the plants might have been seen, and then to search as much of the suitable habitat in that area as we could. But it is certainly possible that we just didn't get to the place where the plants were. So it's very possible that, if we had continued searching on maybe the next property over, that maybe that's where the plants actually were. So poor location information could have contributed to this as well. These surveys were in 2018, which is the year, almost exactly a year after Hurricane Harvey. And we certainly saw, especially on the southern portion of the range, we certainly saw lingering damage from Hurricane Harvey. This particular site, we were told that it was about 13 feet underwater, so 13 feet of water higher than it currently was at the time. And you can certainly see damage to this. Most of the hummocks had been eroded away and washed away. So we certainly had some lingering evidence of Hurricane Harvey. And if we're talking about a tiny little plant, the very shallow root system, that type of flooding could certainly have impacted populations. We saw a presence of erosion on some of these hummocks, where you can see here, this used to be covered in moss, and this moss had been just kind of eroded away, washed away by flooding. So in some of the places where we didn't have firsthand accounts of how much water was there, we were able to see that there was, in many cases, this was a very common thing to see, these kind of washed away moss areas. We also had a number of populations where we were able to see some evidence of changes in hydrology. So this is a plant that occurs essentially in a wetland environment, right? It's dependent on this moisture in these seeps and in these bagels. So if we change how water is moving across the landscape, that can really alter the habitat and make it so the plant can no longer survive there. So we saw a number of places where we thought we had really good location information, and we got to the place where we thought we were supposed to be, and it was completely dry, or there was really the only evidence that there had been moisture there was maybe some lingering magnolias in the canopy, or an area with a bunch of dried up ferns. So we had evidence that there may have been moisture there, and that might have been where the plants were, but the hydrology had changed for whatever reason, whether that be natural changes or man-made changes. So that could have impacted some of these populations. We did see evidence of feral hogs. And if you think about this kind of really wet and mucky environment, they really like that to dig around in the mud. So thinking about this little tiny plant, it certainly could have been impacted by that. Although none of the exact points where we were pretty certain we should have found the plants had evidence of feral hogs, but kind of in the surrounding areas, we did see a lot of evidence of feral hogs. And then finally, we saw at a number of the sites, we saw really a large accumulation of pine needles on the ground. And if we think about the kind of the natural habitat, pine is certainly common in the overall, matrix of the environment at the more upland levels, but down in these bagels, you don't see pine trees. So when we have pine needles filling the bagel and just sitting there and accumulating, that really changes the understory layer. So we don't know what impact exactly that would have, but the evidence that the presence of those pine needles to us is some sort of indication that that we have some disturbance happening in the surrounding landscape, which could be impacting the plants if the presence of the pine needles is not doing so. We didn't find plants at any population that had a large influx of pine needles. So just overall, our conclusions that we came up with, that there were 6 populations as of 2018 in the state of Texas that had plants confirmed. Potentially there are more plants out there, but at that point we only found 6 populations. We concluded that sites that are highly disturbed seem to be less likely to support Texas screwstem. And then those sites that are really high quality, that are really left in pristine conditions, potentially probably do support a number more plants. So I do think there will be more populations found, particularly in areas like the Big Thicket or in the National Forest, these areas that are highly protected. However, I do want to point out that the potential extent of the distribution for this plant is always going to be limited, because it is restricted to this really narrowly distributed habitat. So this plant will never be very common. It will only ever exist in this really unique habitat. So if we lose this habitat, we will lose this plant. So, if we think about what the overall goals of this project were, it was to try to inform the listing decision, right? To try to inform whether or not this plant should be listed as a threatened or endangered species under the Endangered Species Act. So, that's not my job to say whether it should be listed, but I certainly think that with only 6 populations found in the state of Texas, we certainly seem to see some level of decline and only a handful of plants present. To me, it seems like it potentially could warrant being listed. So what can you do to prevent extinction? You know, this whole project is focused on trying to protect this plant. And there's a number of things that all of us can do to try to prevent plant extinction or extinction of really any organism. And the first is really just to enjoy nature, to get out there and enjoy nature and to share that enjoyment with others, because we are not going to protect something that we don't appreciate. So it's important to make sure that we all appreciate the natural world and appreciate all of these, you know, unique and beautiful organisms that we find there. I would really, really love if everybody here would start to use iNaturalist if you don't already. If you are out in nature, you know, take photographs of what you see and share those on iNaturalist, because this is a really great tool for us to keep track of what's going on with the plants and the animals in the state. And, you know, there's only a handful of biologists and conservationists, and in particular botanists in the state. So the more hands we can have out there, the more eyes we can have out there looking for these things, the better chance we're going to have to find them. You can always give your time in the form of volunteering or your money to conservation groups, volunteer for Texas Parks and Wildlife, help out with projects if they come up, work with your local chapters of the Native Plant Society. There's all kinds of organizations out there that work to protect our native plants and animals. And I would encourage you to get involved with those. And then finally, really, in order for us to protect, you know, plants and animals, we have to find a way to live in harmony with them. So helping to cultivate, you know, conservation ethic within our communities, you know, embracing things like sustainable development and local food production is really something that we can all do. So that just leaves us with kind of the final question, and that is why? Why should we protect all of these, you know, interesting organisms? Why should we protect this unique little plant? And, you know, this is a really, it's a difficult question to answer, and it's a question that I get asked a lot. You know, so I could tell you that, well, you know, we all rely on plants. Plants are, humans need plants for food. We use plants for clothing, for shelter. You know, we rely on plants, but that doesn't really hold true for this species. You know, this plant is unlikely to ever really do anything for humanity. So it really comes down to the idea that, you know, all species have a unique evolutionary history. They have their own unique and rich story that that if we lose them, we lose that, you know, and it's a part of our heritage and a part of our sense of place and sense of belonging to a place. And, you know, they have as much right to existence as we do. So for me, it's about the fact that this is a living thing that has every right to live on this planet, as much right as we do. So if we are the ones threatening them, I really think we should be the ones protecting them. So I just want to thank a couple of people. First off, I want to thank Heather Bass, who was the research assistant who helped me with this project. This was as much her project as my project. So thank you, Heather. Anna Strong, the Texas Parks and Wildlife botanist who oversaw this project, the entire non-game and rare species program at Texas Parks and Wildlife and the Texas Natural Diversity Database, for all of their wonderful assistance and help throughout this project. And also, this project was funded by the Conservation License Plate Program. So if you don't have a conservation license plate, I highly encourage you to buy one. The proceeds go directly to funding conservation projects within the state. So thank you to that program for funding this project. And then finally, I just want to thank the landowners and the land managers who took us out and showed us their properties, who let us access their properties, who loaned us their ATVs, and who really are the stewards of these plants on the ground. We could not have done this project without them, and I'm so grateful to all of them for letting us share this with them. So with that, I'm happy to take any questions. I hope we have a few minutes. If you don't have a question today, but you think of a question later, I put my contact information here for you. So please feel free to reach out and send me an email if you have any questions or comments, or if you find a rare plant that you want some more information on. So thank you, and I guess I'll turn it back over to Anna.
Great, thank you, Kim. We do have some questions. So the first one, actually the first two are very similar. So I'll ask them together. Is what defines a population? How big or what is a population?
Okay, so a population, basically when we put all of these kind of points on a map, we looked at their proximity to each other, and we used kind of a separation distance of a kilometer. So anything that was more than a kilometer apart, we considered as different populations. So if things were within a kilometer of each other, we considered that a single population, with the idea being that the genetic diversity within that kind of area would be shared because of pollinators or things like that within that group. So was that the only? Does that answer that question?
No, there's well, that's the only question about populations.
Okay.
The next question is, and I believe you have already answered this question, is there any demonstrated value to the Bartonia that we are aware of?
Demonstrated value to humans? No, but certainly demonstrated value within the ecosystem. You know, we don't know what this plant does within the ecosystem. You know, is it essential to another organism within the ecosystem? But for humans, no, we don't know that it essentially has much direct value to us other than its existence.
Does this subspecies share habitat locale with other subspecies or species? Could some of those collections be misidentifications, or is subspecies texana not likely to be confused with anything else?
Okay. Yeah, I had a huge section in this presentation about the comparisons with the closely related and often overlapping in distribution Bartonia paniculata, subspecies paniculata. But I took it out for the interest of time. Yes, it looks very much like this other subspecies, Bartonia paniculata, subspecies paniculata. They do often occur in the same habitats. We did our very best to look at every single specimen of both of those species to try to determine to make sure we didn't have any misidentifications. So that was something that we spent a lot of time trying to do. But one of the things that the Matthews et al. Paper pointed out was that the characters that define those two subspecies are often overlapping. So we really can't just look at a single character, like the length of the petals, for instance, and know that we're looking at the right plant. So there's still a lot of taxonomic uncertainty, and it is possible that some of those collections could have been misidentified even by us with that second look. Was that all of the question?
Sorry, I
Okay.
hit mute and didn't hit it.
Okay.
Yes, that is all the question. The next question is, are you able to use the latitude and longitude of the pictures uploaded to iNaturalist or the map detail function on iNaturalist?
Yes, so if as a user you allow those coordinates to be shared, we certainly use those. And that is one of the things that makes iNaturalist so valuable to us, is that we can have those coordinates. Now, as a user, somebody uploading a picture, you can choose not to share those coordinates. But I would encourage you to at least allow those coordinates to be shared with the curators of projects, because even if the whole community on iNaturalist can't see those coordinates, as a curator of one of those projects, which most of the conservationists across the state are, we can use that information when a general member of the public might not be able to. So yes, we definitely use those coordinates.
Yeah, so TPWD has an iNaturalist project called the Rare Plants of Texas. Yeah. And if you submit observations to that project, but keep your lat long hidden or obscured, we at TPWD can see those locations. if you add that observation to the project Rare Plants of Texas.
Yes. Yeah. And if I'm going through and I find an observation that is not in there, I certainly try to add it. So if you don't add it yourself, that's fine. Somebody will find it and add it for you.
The next question is, what time of year is best to find the plant?
Yeah, so it blooms, starts flowering in about October, and we were able to find plants up until December. So it looks pretty much the same from October until it disappears. I suspect it's October until we get a freeze of some sort. So probably October to December, so in the fall is the best time to find it.
So this is related to one of our previous questions, but have you considered opening up a project on Inaturalist in these counties to get citizen scientists to help you find additional populations?
I haven't, although, there is the Rare Plants of Texas project on iNaturalist. And I would be hesitant to send people out trying to find this plant. Just because these communities, it's really difficult terrain to get through, and it's really hard to find the plant. And I wouldn't want too many people pushing through these systems. So I would be hesitant to do that for this particular species. I think a species that's a little more showy, maybe in a habitat that's a little less susceptible to damage by foot traffic, I would certainly consider that.
Is Bartonia represented in the seed bank?
Are you talking about the soil seed bank or the conservation seed bank? And I guess I can probably answer both of those. I don't know if it's in the soil seed bank. It is an annual species, and its population numbers do appear to fluctuate greatly from year to year. So I suspect it's present in the soil seed bank, and it may persist for an unknown length of time in the soil seed bank. One of the outcomes of this project was that we were able to actually find a population that was large enough where we could get permission to collect seed for this population. So Mercer Botanic Garden was able to come out and collect seeds at one of the populations to get this species into the seed bank. And as far as I'm aware, that was the first seed collection made for this plant. So it is now represented in a conservation seed bank.
There are also some kudos to you telling you that you're a great presenter and that you answered questions before they were even asked. But there are additional other questions. Any potential for growth in areas closer to the coast, in areas where water is more brackish than fresh, such as Orange County?
I don't know the answer to that. I suspect that the habitat for this species is pretty narrow, in that if we don't have those kind of exact conditions, it's probably not going to be there. And I'm not very familiar with Orange County and kind of what the habitats look like, so I don't know the answer to that question.
Another thank you from Tiana. I appreciate the importance you paid to the contributions that can be made to science at the local level via INAT observations and in sharing one's knowledge. Sue Riley asked, is it possible to restore bagels like other wetland restoration projects where they have been lost?
I suspect it probably is. We saw a lot of evidence of, damage from flooding and from hurricanes, but I suspect that those bagels will regenerate and, those hummocks will reform. So in those instances, I think that, time will eventually restore these bagels. In terms of, human efforts to restore them, I suspect just kind of restoring the natural hydrology of the area would be our best opportunity to do that. So beyond that, I'm not sure, but I'm not a restoration, ecologist. So I certainly can't fully answer that question, but I think that just really time is probably what could restore these bagels.
We are now at 1 o'clock. So Kim, if you have time, we still have more than half of the participants on, and I will just continue until they start to drop off precipitously.
Okay, sounds good.
Or you need to go.
Okay. I have a few minutes. So.
Okay, good. Well, let me know. as an annual plant, do you know if Texas Screwstem has bad and good years in terms of number of plants in a population?
I suspect it does. We found very small populations, but I know there have been some surveys more recently done that have found larger populations. And some of the literature suggests that population sizes do fluctuate, which is really common with annual species. So I suspect it does have good years and bad years. And we may have just been surveying in a bad year for the plant. So hopefully that wasn't a good year where we only found, 60 plants or so. But yeah, I do think that that's definitely the case.
How much is this story the typical story of rare plants in Texas? At some level is hard to find, synonymous with rare.
Yeah, that certainly, plants that are hard to find, or at least for some portion of their history, are going to be considered rare because they're hard to see. But I think when you have multiple efforts, multiple time frames where botanists, who know the plant really well, are going out and systematically searching habitat that they believe would be ideal for the plant, and not finding it, then it truly is rare. And we've seen that with this plant. We're the third instance where the third, targeted survey for this plant, and I believe there's been another one since this survey was started. So I think that is the case. And I think in terms of it being a typical story, just the overall lack of available information for rare plants is kind of the typical story for rare plants in Texas. All of our species need this type of effort on a regular basis because conditions are constantly changing and we just don't know what's happening from year to year. And we can't get out there except for maybe every 20 or 30 or 40 years for each of these species. So it is potentially somewhat a typical story, but I do think for this plant, it probably is, truly is a rare plant.
And what are the benefits of a plant being listed as endangered or threatened?
As a threatened or endangered species, it comes with some federal protection. There's some, money allocated to their protection and to restoration and of their habitat and things like that. Now, for plants, those protections maybe don't extend as far as they do for animals. On private property, there really is no protection for plants, threatened or endangered plants on private property. But in terms of, this plant occurs in kind of this area that is really heavy for forestry, for timber cutting. So helping to protect the habitat would go a long way to protect the plant.
How do these plants reproduce? Do they depend on pollination?
I don't know. I don't know how they're pollinated. I assume they're pollinated by either some sort of insect or wind, and I really have no idea. That's a very good question. And I think this whole genus, I think there's a lot of information that needs to be done, and someone else might know the answer to that question. So if someone does, please jump in on that one.
Yeah, I was reviewing the assessment that I wrote a few years ago, and I couldn't find any information that had been documented definitely on this species, although that may not be the case for other Bartonia species.
Yeah.
Do we know what stimulates germination? How consistently does it emerge at known sites, or do we know?
We don't know. I know that Mercer, since they've collected seed, have started to do some experiments on this. So hopefully we'll have some more information from them soon. But no, we don't know. We really just don't know what causes the seeds to emerge, what causes one year to be good, one year to be bad. Yeah, we don't know those answers.